Today, we're going to go over what whole genome sequencing is in regard to food safety in under 3 minutes. First, every microorganism has a genetic code that is unique to that organism. So in essence, it's like having a fingerprint. So basically, the FDA can help find the origin and cause of a food outbreak by culturing the bacteria taken from a sick individual, then isolate the DNA or fingerprint of the perpetrator, and then have a molecular scissors or enzyme that cuts these up into smaller pieces, and then it gets replicated many times over, which is then loaded onto a sequencer to have its genetic code read, creating a fingerprint library. A specialized computer sorts all these puzzle pieces and puts them back together again to create a whole genome sequence or fingerprint. So how does this help find the cause of outbreaks? Well, when the consumer is eating a contaminated product, they have no idea a disease-causing microbe or pathogen is in there. It can take hours, days, or even weeks depending on the pathogen to see any symptoms. And this could be taking place across several different states at one time, but they're all caused by the same microorganism. It is the responsibility of the FDA and other public health colleagues to keep the food supply safe and find the origin and cause of the outbreak. So if they have the fingerprinted culprit, they can search several different suspected food plants for a match. They can test things like finished product samples or swabs of areas from food manufacturing environments, such as floors and other surfaces. There could be a number of different microbes inside of the plant. Some good, some indifferent, and some that are bad. Some potential sources of contamination in a food plant are people, tools, water, ingredients, product equipment, air, and plant infrastructure. So the FDA can use the fingerprint found from an outbreak and compare them to the bad microbes found in your plant. If it's a match, they have found the origin of the outbreak. The consequences of this is recalls, which are expensive, potential plant closure, as well as having jail time. Yikes. 3 recent examples of recalls that were able to use whole genome sequencing to find the root cause were the El Abuelito recall with fresh cheese, Big Olaf Creamery with ice cream, as well as Old Europe with brie and camembert. To recap, whole genome sequencing can be used to find the source of foodborne illness outbreaks, either by public health partners evaluating clusters of illness, then identifying the source based on those facts, or product or environmental samples found at your facility get linked back to foodborne illness cases due to the fingerprint match in the database. GMPs and sanitation prevent these recalls.